Hi everyone, today I will introduce what is inhibitor to all of you. Today's presentation has four parts. The first is inhibitor introduction. The second is inhibitor classification. The third is inhibitor and targets, common term and parameter. The last is biological research with inhibitor. What is inhibitor? From a broader perspective, inhibitor is a molecule which could reduce or block the activity. From a biological point of view, inhibitor is a molecule that binds to a protein and decreases its biological activity. For example, an inhibitor could reduce enzymatic an inhibitor could reduce enzymatic reaction. We call it enzyme inhibitor. The inhibitor could reduce transcription activity. We name it transcription inhibitor. Both antagonist and reverse agonist are also inhibitors. This picture, this one, is a classical enzyme inhibitor diagram. The red protein is enzyme, the blue one is substrate. They could perfectly bind together through activity site. If the inhibitor has a similar structure with the substrate, it could enter the active site to block substrate entries. So the substrate cannot enter the active site. This diagram, this one, is about an inhibitor of protein-protein interaction. If the inhibitor has a similar structure of red protein's target binding site, it could replace red protein to bind with blue protein. So this inhibitor could block protein-protein interaction. Which kind of molecule could be an inhibitor? Inorganic ions, polypeptide, small molecular inhibitor, and macromolecule protein. All of these could be an inhibitor. So far, in research field, small molecular inhibitor and macromolecule protein are used as an inhibitor mostly. Let's talk about inhibitory antibody, which is a kind of macromolecule protein. For example, the inhibitory antibody of programmed cell death protein 1 could block the interaction between PD-1 with its ligand PD-L1. It could activate the immune system to attack tumors. Now, I will introduce the drug nomenclature of antibody drug. One inhibitor has different names in laboratory, clinical, and market, which are chemical name, generic name, and drug brand respectively. There are some examples. Merck developed a PD-1, PD-L1 inhibitory antibody. In laboratory process, its name is MK3475. In clinical stage, its name is pembrolizumab. In market, its brand is Keytruda. We could find a pattern here. In the laboratory part, the name comes from the abbreviation of company name. MK abbreviates from Merck. BMS abbreviates from Bristol Myers Squibb. CT abbreviates from CureTech. We could find another interesting fact. In clinical stage here, all the names are suffixed with MAD, which comes from monoclonal antibody. So we can conclude that the name of inhibitor suffixed with MAD, means inhibitory antibody. Selicam also provides this kind of inhibitor. So far, we provide 5 inhibitory antibodies. There is another commonly used inhibitor, small molecular inhibitor. This kind of inhibitor is one of Selicam main products. People also call it small molecule inhibitor or chemical compound. Customers interested in these names may be our potential customers. How does a small molecular inhibitor work? This is the ATP binding site of PI3K. The particle structure of watamanin or Ly2940002 is similar to ATP binding structure, so they could enter the binding site and block ATP in the reduce and block ATP and reduce the activity of PI3K. Small molecular inhibitor is similar to inhibitory antibody nomenclature. They have a different name at different stages. The name in the laboratory part, the name comes from the abbreviation of company. If the customer just tell us a name of chemical, how could we know whether it is an inhibitor? Now I will introduce common drug suffix and meaning. The suffix NIB indicates a small molecule inhibitor of kinase enzyme. More specifically, tinib is used for tyrosine kinase inhibitors. The suffix mycin indicates antibiotic or antibacterial inhibitor like rapamycin. The suffix vir is used for antiviral inhibitor. The next topic is inhibitor classification. There are 2 classifications. One is according to the target protein of inhibitor. This is what we talked about previously. The other is according to the combination of the inhibitor with target protein. It could categorize into reversible or irreversible inhibitor. Most inhibitors are reversible. Now, let's talk about common term and parameter of inhibitor or target protein. What is the meaning of universality? Generally, there are many inhibitors for one target. Most scientists will use inhibitors with high usage rate in their project. How to find the usage rate of each inhibitor? We could introduce our website to customer. For example, a customer would like to choose an inhibitor for MEK. We could visit the page about the target of MEK. Then we could put our mouse on the name of product, just like this. A bar chart about publication over the past 5 years will jump out. This data is from NCBI. It is authoritative. It is authoritative. This function will directly to tell you which inhibitor of MEK is popular in scientific research. The next is solubility. Some customers may want to choose an inhibitor with high solubility. Our functional website also could help these customers. We list all MEK inhibitors here. You can click the arrow to sort according to the solubility of specific solvent. IC50. The IC50 is the concentration of an inhibitor where the response or binding is reduced by half. There is a similar term called equilibrium constant. The equilibrium constant of a chemical reaction is the value of the reaction quotient. When the reaction has reached equilibrium, either IC50 value or Ki value, the lower value means the stronger activity of this inhibitor. If the customer asks the reason why the IC50 value they get is bigger than the publication, firstly, don't vary the quality of the inhibitor. Generally, the difference in one order of magnitude is reasonable. As we know, a C-type cell line and incubation time, any of them will affect the IC50 value. EC50. The EC50 is a concentration of a drug that gives half maximal response. If the customer asks the reason why the IC50 value they get is bigger than publication, it is the same with IC50. A C-type cell line and incubation time, any of them will affect the EC50 value. And the next, specificity. Inhibitor only works on specific target with no effect or weak effect on other protein. This is specificity or known as selectivity. High specificity or selectivity can avoid off-target activity and side effect in experiment. What is signaling pathway and signaling transduction? Signal transduction is a process by which a chemical or physical signal is transmitted through a cell as a series of molecular events. This series of molecular events is signaling pathway. How to study a specific protein's impact on biological activity. Scientists always increase or decrease the expression or activity of this protein while studying its function. All life activities of organism are related to protein. There are two ways to achieve the goal of decreased protein expression or activity. One is reducing protein level by reducing mRNA. The other is inhibiting protein activity. Only the amount of active protein is reduced, while total amount of protein has no change. For this method, we need to use inhibitor like small molecular inhibitor. How do customers choose an inhibitor? Some customers prefer to choose inhibitors based on the references. They will directly follow the literature of the compound or even brand of inhibitors. For these people, we could introduce this inhibitor's citation part on our website, like this one. I mean this part. Other customers may compare different inhibitors with different purpose to choose a suitable one for their project. They normally consider from three aspects: universal specificity and activity. How do inhibitors work in biological research? Firstly, scientists use inhibitors to develop the function of a specific signaling pathway or target protein in biological activity. Inhibitor also could be used as a positive control. For example, rapamycin is a classical cell autophagy inducer. Rapamycin is always used as a positive control in autophagy research field. It could be used for inhibitor-reduced animal or cell model. Inhibition of certain targets can induce cell or animal pathological changes that scientists are interested in. These inhibitors can be used to make cells or animal models. It is also used for drug or target screening. Some customers buy a screening library for target screening or picking some compounds of specific biological activity. How to find inhibitor customers? Actually, most laboratories use inhibitor. The difference is just amount. Our aim is to find healthy bears. Which research field will use a large amount of inhibitor? The disease-related research like cancer, immune disease, and so on, stem cell, new drug development, pharmacology, a signaling pathway, and protein structure. All above may need a large amount of inhibitor.